professor of translational neuroscience at Oxford, and I work on cerebellar ataxia here at the Kavli Institute. So cerebellar ataxia is a movement disorder. Patients that have cerebellar ataxia have difficulties with walking, with keeping their balance, controlled movements and coordination. Patients often have difficulties with their speech, so they often have a slurred speech. They might have trouble swallowing, and they also have problems with their eye movements. So usually cerebellar ataxia arises from damage to the cerebellum, which is a part of your brain. It's actually this really fascinating part of the brain here, the brown bit at the back of the brain, which lies back here. There can be many different causes for cerebellar ataxia, for example, a tumor or a stroke, but we are particularly interested in gene mutations that cause cerebellar ataxia. Cerebellar ataxia can be a very debilitating disease that affects patients' quality of life and also can lead to early death. Ataxias are rare diseases, so in the UK there are approximately 10,000 adults and probably around 500 children that are diagnosed with cerebellar ataxia. So it's a rare condition, but very similar to similar neurodegenerative disorders, for example, Huntington's disease or ALS. Importantly, there's currently no cure or effective treatment available to patients with cerebellar ataxia. My group is really interested in understanding the underlying genetic and molecular mechanisms that cause cerebellar ataxia. So we have been involved in gene discovery around cerebellar ataxia. So we have identified 2 novel genes that, when mutated, cause the disease. And now we're really interested in understanding of why is it that mutations in these 2 genes cause this particular disease. We specifically focus on Purkinje cells, which are particularly affected in cerebellar ataxia. And ultimately, we would like to use the knowledge that we gain from our studies to design better therapies or more effective treatments for patients. We also develop novel models to study cerebellar ataxia. And so, for example, we are developing human cerebellar models, differentiating nerve cells as well as 3-dimensional so-called organoids of the cerebellum that we differentiate from induced pluripotent stem cells to have better human models available in the lab that we can use to understand the disease and ultimately test treatments. So one of the challenges in ataxia research is the vast heterogeneity of the disease. So the clinical picture can be very different for different patients, and we also know that there's a lot of different gene mutations that can cause cerebellar ataxia. So for the dominant cerebellar ataxias, we have more than 30 genes, and for the recessive forms of ataxia, there's even more genes that are involved. And this makes it really difficult for us to understand what is really driving the disease and what are the mechanisms that we could target for specific treatments. Also, cerebellar ataxia is a rare disease, so we don't have that many patients, and again, that makes it difficult to work on this disease. Another challenge is that we have a lack of non-invasive biomarkers that we could use to track the disease early and also to use to see whether treatments are effective. And again, I think that will be a really exciting development in the near future that we can use, for example, imaging or other methods to see or to catch when people are affected early and then to help them. So I'm really excited to be part of the Kavli Institute and really excited to work together with colleagues, use their expertise in addressing some of the questions that we are interested in. And we are already working together with some of the groups in the Institute to use technologies that we haven't used before and that could help us to better understand cerebellar ataxia and to form new collaborations.